So let's introduce our next speaker, Professor Leyla Isik. So Leyla is the Clare Boothe Luce Assistant Professor of Cognitive Science at Johns Hopkins University and holds appointments at the Department of Biomedical Engineering and Kavli Neuroscience Discovery Institute. And before joining Johns Hopkins in 2019, she was a postdoc at MIT, actually, and Harvard in the Center of Brain, Minds, and Machines, where she worked for Nancy Kahnweiser and Gabriel Kremen. And she received her PhD from MIT, advised by Tommy Poggio. So welcome back.
Thank you.
Her main research interests are in understanding the cognitive and neural basis of dynamic social vision and how we might implement these abilities in AI systems. And her work has been recently supported by Google AI Faculty Research Award and has been published at prestigious journals, including PNAS, eLife, Nature Communications. And from a personal perspective, I want to say that her work is very seminal for the lab here because she was the first to publish a journal article with data from our facility. So that's a very special title. That is a wonderful paper that she published back in 2014, so I recommend for everybody to read The Dynamics of Invariant Object Recognition in the Human Visual System. You may be presenting some of this work here.
You're going to hear a little bit about it.
And this, after this work was published, we had a sequence of other wonderful articles from different groups here in high-profile journals as well. So her lecture today is titled From Neural Dynamics to Neural Computations, Recognizing Objects, Actions, and Social Interactions. Thank you, Leyla.
Thank you, Dimitrios. And thank you to McGovern and Megan for sponsoring this and inviting me. It's always really fun to be back here. So we can very easily recognize all sorts of rich information from our visual system. So this is a video of my son Rami on the left and his friend Sylvia on the right. And before I even play it, you can recognize all sorts of things. They're kids, they're outside, you might see their baskets, and maybe you're checking out Rami's cool sunglasses. And when I play the video, you can also very quickly recognize all sorts of things. So even though I'm not giving you any sound, you can tell that they're communicating. You can tell they have a goal to open that Easter egg. And you can tell that they're helping each other. You can also make rich inferences about their mental states, like why are they trying to get in this egg? Maybe there's candy inside. Maybe he's going to eat that candy, or maybe he's going to share it. And so the overarching question we try to get at in my lab is how do we do this? So this first group of properties, not to say that they're solved, but I think we have a pretty good neurocomputational framework for how we recognize single people and objects. And the focus of my lab is really on the second group of things, so how we recognize people's actions, goals, and social interactions. And these processes are separate from that last group where those sort of mental state and trying to answer those why questions. And we argue that they're much more similar to that first group of properties. We call these different aspects of social vision. But compared to other aspects of visual perception, they're much less well understood. So the approach we try to take in our lab is to take the methods and tools that have been helpful in helping us understand visual object recognition to understand different aspects of social vision. So in particular, we use a combination of high spatial and high temporal resolution neuroimaging, behavior, computational modeling, and critically, we try to compare the representations across all of these different modalities. And so today my talk is going to have two parts. In the first, I'll tell you about some of that work Dimitrios mentioned, where we tried to disentangle the different computational processes that underlie invariant visual recognition using a combination of MEG and computational modeling. And then in the second part of my talk, I'll tell you about more recent work where we try to get at the computations underlying different aspects of social scene processing using a combination of fMRI and EEG. All right, so to recognize objects, like Rami's sunglasses, you have to develop invariances to all sorts of different transformations, right? So even if I show them at different sizes, viewpoints, positions, you can tell that these are all the same pair of sunglasses. So how do we do that? Well, we know that this type of information is represented along the primate ventral pathway, which I'm highlighting here in red. And we know from physiology recordings usually in single areas, that in V1, neural signals reach this region around 40 to 50 milliseconds after an image is shown, and that these cells are mostly tuned to simple features like oriented edges. In primate IT, there are representations for whole objects that tend to be able to generalize across many of those transformations I mentioned, and that takes longer, more like 100 to 150 milliseconds for signals to get there. But the timing and processing stages in humans was difficult to disentangle. From ERP measurements, we knew that by around 150 milliseconds, a lot of invariant high-level information was represented. But we wanted to ask if we could read out earlier stages in the human visual brain, and if we, in particular, we could tell when neural signals could generalize across these different transformations. So like Demetrius said, we use the MEG system downstairs, and most of the work I'm going to tell you about today uses a decoding approach. So we take the activity, the time-resolved activity from all of the sensors, and at each time point, we fit a linear machine learning classifier on one subset of data and try to predict what the subject was looking at. And we use the accuracy of the prediction to assess what information is present in the neural signal, and with MEG, we can do this in a time-resolved way. So as a first step, we just showed a bunch of different image sets to subjects. So for example, these grayscale rendered 3D objects. And I can plot for you the classification accuracy versus time. And so before the image goes on, this is a 25-way classification. We were at chance. And then pretty quickly, you see a dramatic rise in decoding accuracy that starts here around 60 milliseconds. But what we really wanted to understand was when these neural signals could generalize across different transformations. So to do that, we can take a subset of these objects and show them at different positions and train our classifier on the MEG signals from people viewing an object at one position and test at a second position. Now, if we do that, you can still decode, but the signals come on quite a bit later. So this blue bar at the bottom tells you when we're decoding significantly above chance. And so compared to the 60 milliseconds we saw before, this happens quite a bit later, after 100 milliseconds. And we can do this for every combination of positions that we showed the subjects and scales. And what we see is that invariance seems to arise in stages. So pretty quickly you can decode signals within one position or size. But then to decode across different sizes, the decoding latency corresponds to how large a transformation you showed. So you can decode across a 1.5x scaling much sooner than you can a 3x scaling. And the same thing is true for changes in position. And this was nice because this gradual buildup of invariance mimics what we know happens in convolutional neural network models, where there is local pooling in position and scale to build up invariance over larger and larger regions of an image. And so we showed this with an earlier CNN, the HMAX model, that there was a good correspondence between the pattern of MEG decoding and the model's outputs. This has also been shown by many people later with more modern CNNs. And we're now looking at it in more naturalistic scenes with the wide array of deep networks that are available now that I'm happy to chat a little bit about later if people have questions. And so what we saw is that as you go through the first 200 milliseconds after you see an image, you can we first have representations for objects that are not invariant, and then gradually get more and more invariant. And this mimics what we see in early and late layers of a convolutional neural network. And for us, this was a really nice first proof of principle as well, that you could differentiate these different computational steps in the human brain using MEG decoding. So next we turn to action recognition. So for example, trying to open this egg. And action recognition poses many computational challenges beyond object recognition, right? So you often need dynamic information. And actions are also subject to a wider array of transformations than objects. So you have the sort of affine transformations that I just told you about, like changes in position and scale, as well as others. And what's special about affine transformations is that they preserve the relationships between points, lines, and planes. And As a result, all images transform the same way if we're talking about affine transformations. In contrast, there are non-affine transformations like changes in viewpoint, and then several that are action-specific, right? So it depends on the individual person that you're looking at's body, right? An adult and child move in very different ways, et cetera, as well as the pose of the body, et cetera. And so to do this, we designed a data set of people performing different actions from different viewpoints in a naturalistic scene, but one that was still controlled. And we showed subjects in the MEG different actions. Again, they saw all of these video clips. And as before, we tried to decode both within and across viewpoint and also within and across actor. And so what we saw is that within viewpoint, within about 200 milliseconds, we could tell what action somebody was viewing. And so next, we really wanted to try this across view case to see when you could generalize across this pretty complicated transformation. And one hypothesis might be that because this is a pretty complex problem, it would happen later. But in fact, what we saw is that this happens at pretty much the exact same time. I can even zoom in and show you what that, those two traces look like within that critical 200 to 500 millisecond window, and they're really right on top of each other. So what we saw is that in contrast to sort of this gradual buildup of invariance for objects, for actions, within 200 milliseconds, you can recognize that action, and your representations are already viewpoint invariant. This is in contrast to what others have shown in physiology recordings in IT for faces, where you gradually build up invariance to viewpoint, and what we saw for objects in our MEG data, which suggests that there's something different about the computations used to recognize objects that's not common in basic face and object processing. Despite these differences, however, we saw that the same type of convolutional neural network model could match the MEG data if we extended it in a few key ways to make the convolutions happen in 3 dimensions, so in space and time, and we integrated viewpoint invariance in a certain way. And so we, I'm not going to really have time to talk about the details, but we developed a new 3D CNN that not only did a good job at recognizing actions in held out videos, but also provided a good representational match to our MEG data. And so this work sketched out the first couple of 100 milliseconds of visual processing for both object and action recognition. And I think by having this detailed timing information, it allows you to constrain your computational models in a way that you can't do with high spatial resolution data alone. So in the second part of my talk, I want to tell you about more recent work from my lab focusing on EEG. Sadly for us, we don't have an MEG at Hopkins, but I hope you'll still stick around and hear something about that work. So in order to get a rich understanding of this social scene, we have to move beyond just recognizing single entities, like individuals, objects, and individual actions. And so a big focus of work in my lab now is on recognizing other people's social interactions, like the fact that these two are communicating and helping each other. And a big question we try to ask is, to what extent are these types of higher level information also extracted via the same types of bottom-up hierarchical processing that we use for objects? And so in a recent TICS paper, we argued that yes, that these type information still is extracted in a similar visual hierarchical fashion for not only these features that we called visual social primitives, like whether two people are close to each other or facing each other or moving contingently, but also for different types of social interactions, like people who are engaged in a physical interaction like dancing, communicating, and people helping each other or not. And so to get at these questions empirically, you really want both high spatial and high temporal resolution data. So now I'm going to tell you about 2 studies that use EEG fMRI fusion, adapted from a method developed by Radoxici, Dimitrios, and Oliva. And so for this, you can show the same images, often to different subjects, but the same set of images in both fMRI and or MEG. And what you do is you look at the representational space of these images in a given region in your fMRI data, and at a given time point in your M or EEG data. And you correlate these two to get a high temporal resolution picture of how information is changing in a given region in your fMRI data. So in a first study along these lines, we looked at how people process social interactions in controlled stimuli. And this is work led by a former postdoc in my lab, Hamy Limason, who just started her own lab at Durham University. So first with controlled stimuli, I can show you a clip like this or like this. And you can tell they have very different affective content. And so in her prior work, Hamy showed that the information about the social nature of these scenes is represented not only in regions that we typically associate with different aspects of social perception, like the temporoparietal junction and the posterior part of the STS, but also in somatosensory cortex, and that when you view these scenes, these two regions are functionally connected, suggesting that perhaps the way you recognize the social information is from top-down simulation signals in somatosensory cortex. However, it's impossible to tell the direction of information flow, or very difficult to definitively do this with fMRI alone. And so we took these same video clips and showed them to subjects in EEG and tried to get at this question using two different approaches. One, to look at the processing speed, so how quickly you can tell the social content of these videos, and then also try to link the EEG data to this fMRI data to tell which direction this arrow goes for the information flow. So we used these stimuli created by Hamy and Hans Optebeek that consisted of a bunch of different types of affective contact between either pairs of people and also matched actions between a person and an object. So it could either be two people hugging or something that's matched for biological motion, like somebody picking up this box. And so we showed shortened video versions of these video clips to subjects in the EEG and did a similar time-resolved decoding. But then here we use the decoding accuracy as a distance measure to do time-resolved RSA. So at each time point, we make a stimuli by stimuli matrix, which shows the pairwise decoding between every pair of stimuli as our distance metric. We can also get a bunch of labels for these videos, right? Either through automatic methods for labeling visual features like deep neural networks, or from subject ratings for how social the image is, the valence, and arousal. And we can use the Euclidean distance in these feature spaces to compare to the EEG data at each time point. So if I do this, for example, for an early layer from a deep neural network, what you see is that pretty quickly, around just under 100 milliseconds, I'm showing in blue, the representation of the DNN correlates really well with the EEG signals. This is also true for the biological motion in the videos and the raw motion energy. And so between 100 to 150 milliseconds, you get these visual representations from the videos, which was not surprising. But then we also looked at the social content in the videos, so whether it was a video of two people hugging or someone picking up a box, the arousal and valence. And what you see is that within 200 milliseconds, you also have representations of all these pretty high-level social features, suggesting that these social affective features are represented just shortly after the kind of more basic visual processing. We also wanted to, like I said, look at the direction of information flow across these key ROIs. So we took the fMRI data from Hamy's prior study, where she looked at early visual cortex, a combined ROI that includes both the TPJ and PSTS, and somatosensory cortex. And you can take the voxel-wise patterns in each of these ROIs to get a representational dissimilarity matrix, and like I said, correlate that with the EEG data at each time point. And what you do what you get when you do that is you see that information gets to early visual cortex around 50 milliseconds, to the TPJ and PSTS around 110 milliseconds, but to somatosensory cortex not until about 190 milliseconds, suggesting that information first comes online in early visual cortex, then TPJ and PSTS, and then only later in somatosensory cortex. However, one thing that Hamy's work showed before is that the information in TPJ and somatosensory cortex is very correlated, right? They're functionally connected. And so what we really wanted to do was get a picture of how the unique variance evolves in each region, where you remove this correlated activity across ROIs. And so here I'm going to show you the unique variance of the EEG signals explained by each of the 3 brain regions: early visual cortex, somatosensory cortex, and PSTS, removing shared variance across ROIs. And so when you do that, what you see is that, again, information is first processed in early visual cortex, and then TPJ, and then later, actually, interestingly, it goes back into EVC, suggesting maybe some sort of top-down information to early visual regions. But critically, what you see is that there's no unique variance in the EEG signals that's explained, that the activity in somatosensory cortex explains. And so this really suggests that in these sorts of simple social scenes, social interactions are processed within the time frame of what we think is feed-forward processing, so roughly the first couple hundred milliseconds of visual processing. Although I'm going to get to this a little bit later, but some of my prior work with Dimitrios and Nancy suggested that in more complex natural scenes, this might happen later. But we saw through that EEG fMRI fusion that this information is extracted through what we think of as being social perceptual regions, where it first goes to EVC and PSTS, and then that later involvement of somatosensory cortex is not explaining unique variance in the early EEG signals. And so this and other work has suggested that there is a key role, a key pathway from visual cortex through the STS that is engaged when you process social scenes. And this and other work has led to recent theoretical proposals from, so for example, this one's from David Pitcher and Leslie Ungerleider, that in addition to the 2 classic visual pathways, the ventral and dorsal streams, we might have a third visual pathway along the lateral surface of the brain that's specialized for processing social motion. And so they suggest that this pathway extends from V1 through MT and the STS, and again is specifically tuned to recognizing dynamic social information. They also suggest that this pathway is organized hierarchically, so where, just like we think happens with the ventral stream, where information flows from one brain region to the next in a largely feed-forward way. And so one way this manifests in other pathways like the ventral stream is that you start with small receptive fields in early visual regions that are selective to very simple features. And as you move more anterior, receptive fields get larger and the tuned stimuli get more and more complex until, again, you have high-level feature representations that are pretty invariant in the most anterior regions. So in a first fMRI study, we wanted to ask if you see a similar organization. And so if the features of social scenes are computed in a similar hierarchical fashion. And this was work led by Emily McMahon, a PhD student in my lab. And so she looked at representations in early visual cortex, motion selective region MT, and the posterior and anterior STS. These were all the regions proposed by Pitcher and Ungerleider. But in addition, we also added some other ROIs, the extrastriate body area, which has been shown to be tuned not just for single bodies, but also pairs of bodies and their relations. And also the lateral occipital complex, which again responds to objects, but also to represents scene and object information related to action. So for example, if an action is directed at an object or not, that's been shown to be represented in these regions. And so to comprehensively test this wide swath of cortex, she curated a data set of naturalistic videos that all had 2 people and were 3 seconds long, and played them to participants in the fMRI without sound. She also got a bunch of behavioral ratings online for these features of interest, so whether a scene takes place indoors, whether it's in a big spatial expanse or a close-up scene, whether there's an action directed at an object. These social primitive features, like whether people are close by or facing each other, whether there is either a physical or communicative social interaction, and affective features. We also got automatic visual features like low-level early layers of a convolutional neural network and the output of a motion energy model. And so I'm happy to talk more about this later, but I'll I'll spare you all the many regressions that we did and just walk through the results schematically with you. So what we found is that in early visual cortex and MT, you find unique variants explained by these low-level visual features, as you might expect. In these mid-level regions, LOC and EBA, you see representations for these kind of mid-level scene and object features, and also these social primitive features. And most strikingly in the STS, what you find is that there is unique selectivity in particular for whether two people are communicating or not. And again, these videos are all played without sound. And so this really suggests that, in particular, recognizing communicative interactions is a key goal of this pathway. Here are the whole brain results for one subject. And so each voxel is color-coded by the feature that explains the most unique variants. And you can see that as you move from early regions, you get voxels that are best described by these low-level visual features shown in grays, and then these kind of more purple-yellow mid-level features. And again, the most striking thing is really the STS is pretty much just explained by these social interaction features, and further analyses showed it's really communication. And so as you move from more posterior to anterior regions, just like in the ventral stream, your representations get more and more abstract. And this is one key hallmark of a hierarchy. So we suggested that perhaps as the theoretical proposals posited, these regions are hierarchically organized. But it's difficult to just tell from this spatial information alone if the information is in fact extracted hierarchically. So now what Emily's doing is she is showing shortened versions of these video clips to subjects in the EEG. And so she has shortened them to about 500 milliseconds, and then she can do the same sort of combination of EEG and fMRI data. And so she just finished data collection, so I'm just going to show you a few snippets of analysis that we've done so far. But what you see is that as you go over the first couple hundred milliseconds of processing, the signals move from early visual cortex to more and more anterior regions along the STS. The other thing we looked at was the time course of the different feature representations. And so, for example, I'm just going to show you a few features here. At about 100 milliseconds, you can tell whether a scene takes place indoor or outdoors. You can tell whether people are close by or far apart slightly after that, and whether an action is directed at an object slightly after that. We can also significantly tell if an action involves, if a video involves a communicative action, and if it's high or low arousal, and these both happen later. Although critically here, unlike in the study I showed you before with very simple stimuli, these high-level social features now happen, about 300 or 400 milliseconds after the video starts. And so I think this might be a key difference between more controlled and more naturalistic studies that highlights the importance of looking at this information in more naturalistic studies. And this work is still ongoing, but I'm hopeful also that by correlating these feature representations with the different ROIs, we can get a more concrete picture of, in fact, whether this information is being extracted in a hierarchically. Although temporally, at least, the order of the features are being hierarchically extracted. And so to sum up this part of my talk, I showed you that when you observe other people's social interactions, this information is automatically processed, and at least in simple scenes, it happens very quickly. It seems to be slower in natural scenes. And so this is also consistent with prior work I did with Nancy and Dimitrios that showed a very similar latency and more, in less naturalistic, but still natural short images. But despite this slower latency, we still see that the feature extraction follows a similar spatial and temporal hierarchy that goes along the lateral visual pathway. And so I think all of this is helping us work towards a neurocomputational framework of how you might recognize social interactions. So this is something we outlined in the TICS paper, that as you see a scene like this, we know that there are brain regions dedicated to recognizing people and their bodies and faces in particular, and that this information might be fed into regions that are nearby that seem to also process the relational versions of this information, like whether they're close by or facing each other. This information is then fed into more anterior portions of the STS that represent whether there is a social interaction or not. And in work I won't be able to talk about today, we have developed a computational model that's a graph neural network that uses relational processing to recognize this information from visual input in these types of videos. We think all of this is primarily happening along the STS in what might be a new visual pathway. But of course, it interacts with regions that do things like mental state processing, decision-making, and language. And so a big focus of our upcoming work is going to be trying to link these, what we think are largely visual representations for recognizing social interactions, to these higher-level cognitive processes that help you decipher what's going on in the interaction itself. And so I will end there. I'm happy to take any questions. And so I'd like to thank my lab, especially Hammy and Emily, who did the work I talked about today. And thank you.
Thank you so much, Leyla. We do actually have time for some questions. If anybody wants to ask any questions.
Have?
Can you hear me from here?
Yeah.
I wanted to ask, this was a great talk. I wanted to ask you about whether you guys have any hypotheses that you are testing or planning to test about potential differences that happen along the STS, because that's quite a long stretch, right? That's coding for social information, and I wonder how you're thinking about potential structure within that.
Yeah, great question. So so one, let me just clarify one thing, which is that we are looking, this is obviously plotting all the reliable voxels in this subject, but in particular in our ROI analysis, we looked for voxels that respond more to interacting than not interacting point light dyads. And one thing we've been finding is that that pulls up in almost every subject, actually two distinct clusters, one in posterior and one in anterior STS. So far, we can't really tell anything that is happening differently in those two parts of the STS, despite the fact that they're pretty far apart anatomically. But one thing we're doing now is trying to do more data-driven versions of these analyses. So subjects saw 250 videos, and we can put the responses of the entire STS to those videos into something like some sort of component analysis. And so far, we're still pulling up kind of two big clusters, one in posterior, one in anterior. So I think it's just that right now we don't, but we are seeing differences there in the more data-driven approach that we didn't see in this feature labeling approach. And so one way that we're starting to think about it is maybe if we just need, I'm hopeful that with both richer and larger data sets and more data, that we can start to tease apart these differences. Thanks. Yeah.
In this result, do you see that the ventral pathway actually encoding more information about scene and objects?
Yeah, so do I have the results here? I probably don't have them here, but the main thing that the ventral pathway is responding to are either the scene and objects or the more early regions of the ventral pathway respond to the features from the AlexNet deep neural network, the low-level features from that as well. You see some pretty weak responses for some of these other features, including like whether people are communicating. But I think that's also just a side effect of the fact that these are natural videos and all of those things are kind of correlated. But the much stronger representation in the ventral pathway are these scene and object features. Yeah. Yeah.
For the EEG, do you get spatial specificity? I mean, there are lectures that are more helpful than others. And are you doing the coding just with the raw EEG? Are you doing this in frequency space or?
So everything I've showed you today is just doing it on the raw time series data. In the EEG data in particular, we have played a little bit with trying to either a priori just select certain groups of sensors, and that it's mostly in the occipital and temporal sensors. But for the analyses I showed you and what we decided to do after some piloting is just put everything in. And in the first study, it was just a straight RSA, and that still worked pretty well. So every sensor was weighted equally, and it still works pretty well. In this later study, we are doing a linear regression so that in the training learns to weight the sensors differently. Yeah, Santani.
So
Yeah.
feature, and it feels like, if I'm getting this right, like the AlexNet is just by itself, right? You're not guessing out some other such problems.
So we did this all, so what I'm showing you here, I glossed over all the details so you didn't miss it, is we did a variance partitioning approach. So you can either just fit each of these features as a direct regression to the voxel activity, but what in those results we're doing instead is you can, to like say you want to get the unique variance explained by social primitives, it also correlates with these other features. So what we do for each feature is fit an encoding model with all features less that feature and take the difference. So in this result I'm showing you here, we are accounting for those correlations. If instead you just do the regression and plot the strongest feature, even though you'll still get, since they're all correlated with each other, you'll see AlexNet explaining responses almost everywhere. But if you plot the most predictive feature, you see something that looks very similar. So we did kind of both versions of that.
Okay, since there are no more questions, let's thank Leyla one more time.